today I will discuss with you some gene therapy and genome editing strategies that can be applied for the treatment of beta hemoglobinopathies. Hemoglobin disorders basically affect the production of hemoglobin, which is the protein which is very abundant in our red blood cells, and this protein is necessary to deliver oxygen to all our tissues, so it's fundamental to provide energy to our organism. And these disorders are actually very common disorders, which are basically present all over the world, with more than 300,000 children born each year with a severe hemoglobin disorder. Basically, I'm working on beta hemoglobinopathies in which the gene which is affected is the beta globin gene, and this gene is necessary for the production of the beta globin, which is actually forms together with the alpha globin, the hemoglobin, the real hemoglobin tetramers, and this happen in adult cells, while during the fetal life, so before birth, we have in the blood a different kind of hemoglobin, which is the so-called fetal hemoglobin, which is formed thanks to the presence of the fetal genes, which actually allow the production of the gamma globin gene, which form the fetal hemoglobin tetramer. And just another piece of information that can be useful to follow until the end of this presentation is that so basically these genes have to be expressed at very high level because we have like grams of hemoglobin in our blood, and this is the locus in the genome, on chromosome 11, where these genes are located. But to have a high activity of this gene and so high level of hemoglobin in our blood, it's necessary the presence of this region, which is called LCR, which is fundamental for the activity of this gene. As I told you before, beta thalassemia is one of the actually beta hemoglobinopathies which affect the beta hemoglobin gene, and actually in this case there have been more than 200 mutations which cause a reduced or absent synthesis of the beta globin chain of the beta globin, and in this case, so what we have is that we have a reduced formation of hemoglobin, so these tetramers. Therefore, we have several alpha globins which are actually cannot be incorporated in the hemoglobin. While in sickle cell disease, actually we have normal level of this beta globin in our blood, in the blood of this patient, but actually this is a mutant beta globin, and this mutant beta globin actually can cause a very severe clinical phenotype. Just to explain you a little bit better, actually in beta thalassemia, so we have a reduced or absent synthesis of beta globin, and this alpha globin, which is not incorporated into the hemoglobin tetramer, actually damaged the cells, damaged our red blood cells. And so basically these red blood cells, which are coming from stem cells, they can be produced by in the body of thalassemmic patient, but they actually after a while they start dying. And this causes basically a reduced number of red blood cells and a reduced hemoglobin concentration in our blood. And so the clinical phenotype is basically a typical anemia. In sickle cell disease, actually we have a mutant hemoglobin which is present in the blood, but this hemoglobin stands for some aggregates, and these aggregates actually cause the formation of abnormal red blood cells. So usually our red blood cells are biconcave and round-shaped, but actually these red blood cells in sickle cell disease adopt this sickle shape, and actually they cannot flow freely in the vessels, so they tend to obstruct vessels, and this causes vasocclusions and several clinical symptoms. If you think, for example, that this can happen in the brain, in the capillaries of the brain, the consequence will be like a stroke. Moreover, these patients are also anemic because actually these abnormal red blood cells are very fragile and are recognized by the organism like abnormal, so we destroy these cells. So the patient's organism destroy these cells. So the total number of red blood cells in the blood of this patient is lower. And so we have an anemic phenotype also in this case. Then the therapeutic approach, the main therapeutic approach is basically to give to the patient normal red blood cells via transfusion. But this is not a definitive therapy because our red blood cells actually cannot live, can live just for a certain amount of time in our body. So we have to continuously provide normal red blood cells to this patient, which means that this is a lifelong treatment. And there are also several pharmacological treatments, but these are also for sure not definitive, and moreover they are not effective in all the patients, and they have many side effects. Therefore, of course, the only definitive therapy would be the bone marrow transplantation, which means that we get rid of the hematopoietic stem cells of the patient, which are genetically defective, and then we replace them with the hematopoietic stem cell coming from the bone marrow of a normal donor. And then these hematopoietic stem cells, so in the patient, this normal hematopoietic stem cell will give rise to normal red blood cells and fully functional red blood cells. But unfortunately, this therapy is available only for patients who have a compatible donors, which are unfortunately less than 30% of the patient. So what we and others have proposed is to basically take the cells, the hematopoietic stem cells from the patient, genetically modify them to correct this genetic defect, and then reinfuse them into the patient. So how we can modify these cells? We can use lentiviral vector, so viral vector, to deliver the correct gene into the hematopoietic stem cell of the patient. But recently it has been proposed also to use genome editing tools to reach this aim. And just a few slides about lentiviral vector, which is the strategy which is currently used also in clinics. Actually, we have developed this viral vector, which are inactivated, in which basically we insert the correct beta globin gene, and that is then delivered into the hematopoietic stem cells of the patient. So the concept is pretty simple. So I just want to point out that, as I told you before, we need this region in the genome, in the endogenous genome. So in our cells, we need this genomic region, which is called LCR, this large piece of DNA to enhance the beta globin expression. Therefore, into this lentiviral vector, we don't put only the beta globin gene, but we put also these some fragments coming from this LCR, and we can have to choose just a small fragment because this is a very large region in the genome, and unfortunately we cannot fit it entirely in the frame of a lentiviral vector. So what we did a lot of study in order to define a mini LCR, so a short region which is able to increase beta globin, the activity of the beta globin gene than in patient cells. And we are trying to, in this way we try to get beta globin expression level which are as much as possible close to the endogenous level of endogenous beta globin level. But this is not always an easy task actually. But actually with this therapeutic approach, what we have shown is that basically we were able to ameliorate the thalassemic phenotype and the sickle cell phenotype in vitro, in human cells, in cells coming from the patient. So these are basically thalassemic cells in which we do not observe the formation of red blood cells, which are this round one, because actually the cells die. But upon treatment with lentiviral vector, we saw the formation of red blood cells. So we ameliorate the phenotype. We were able to produce red blood cells. And similarly, in sickle, when we use the sickle cell disease patient, actually we we noticed that upon treatment with our lentiviral vector, there was a partial correction of the sickle cell disease phenotype. And this is, we measure it by basically counting the cells which sickles with this strange shape, which are all of them in the case of the untreated samples, while we saw around 30% of round-shaped normal red blood cells upon treatment with lentiviral vector. And these are basically the gene therapy trials which use lentiviral vector for the treatment, and most of them are ongoing actually. And these gene therapy trials basically are ongoing, and they aim at treating both beta thalassemia and sickle cell anemia. And these are the result of the first patient that has been transplanted at the Necker Hospital by Marina Cavazzana. And actually, this is after 12 months after the transplantation, basically we observe an increase in the levels of hemoglobin. This is a thalassemic patient who had very low levels of the hemoglobin. And actually, after 12 months, this patient is basically transfusion-free, which means that we had an increase in the hemoglobin levels, which are not as high as the one that normal individuals have, but still this patient does no longer requires transfusion. And now, actually, this month is more than 70 months after the transplantation and is still transfusion-free. But actually, so in conclusion for this part, regarding the efficacy of this approach, actually it has been reported the transfusion independence of several thalassemic patients, but I have to say that most of them are like this beta plus thalassemic patients, which means beta plus means they have still some residual beta globin expression, so they're not completely it's not completely zero. But what we think is that it may be necessary, especially for beta 0 patients, which are patients in which we don't have any beta, or for sickle cell disease, we think that we may need to have a higher beta globin expression derived from our vectors, or from whatever approach we want to use, in order to correct this phenotype. And we are still working in optimizing lentiviral vector, of course, but actually another strategy that I will present you in the next slide is a gene therapy, a genome editing based approach, in which we want to modify the endogenous locus and take advantage of the endogenous regulatory regions, such as the LCR. Regarding the safety of this approach, I have to say that lentiviral vector integrates semirandomly into genes. They can integrate into genes, so they, in theory, they can deregulate genes, which is not good if you think that about oncogenes or genes involved in cancer, whatever genes. But actually, especially with this new generation lentiviral vector, these are pretty safe. And so far, many patients that have been treated also for immunodeficient patients, like in Milan, actually there were no adverse event has been reported with lentiviral vector, I have to say. And then, so how we can use genome editing approach to improve the phenotype of this patient? Of course, the ideal strategy would be simple to correct the beta globin the beta globin mutation. And for example, this is the endogenous locus. So we have basically, for example, the sickle cell disease mutation, and we basically just fix it. And this is very easy, but pretty tough, I have to say. But another approach that we are also working on is actually to increase fetal gamma globin levels. And this is because basically in this patient, the fetal gamma globin is not expressed, is not present in our blood in the adult life. But actually in this patient, the gamma globin gene have no mutation. So in theory, the idea is to reactivate them, so activate this gene in order to increase fetal hemoglobin expression. And this is because it has been shown that there are few patients that for a number of reasons, I'm not going to go into the detail, basically they have a persistent fetal hemoglobin activity and expression in the adult life. And these patients, many of them, they cannot be called patient because actually their clinical phenotype is basically normal. They are normal, and or at least there is a really huge amelioration of the clinical phenotype. So to do this, we have to use, we and many other groups are using genome editing tools actually to edit the genome, so to change the genome. And in the last 10 years, there are several classes of genome editing tools have been developed, such as Zinfinger and Talen, which are actually proteins which can bind a specific site on the DNA. And thanks to their domain, this one in yellow, for example, for the Zinfinger, and this one in red for the Talen, which actually can target specifically a region in the genome. Moreover, they have also another domain, another part of this protein, which is the nuclease part, which is basically, these are basically some molecular scissors which are able to cut the genome, the DNA. And then, lately it has been shown that the CRISPR-Cas9 system has been developed, and this is basically, the concept is more or less the same, but I have to say that this system has really brought a big revolution in this field, because it's very easy to use and it's very flexible. And basically it relies on the presence of this protein, the Cas9 in light blue, which can cut the genome. And the difference with the other system that have been developed before the CRISPR-Cas9 is that actually by itself this protein cannot bind directly the DNA, but it needs a small molecule which is called guided RNA, which actually guide and target this protein to the gene. And then these are, so basically you can use these molecular scissors to generate cuts into the genome, and then these cuts are then repaired by the cells, and they can be repaired in different ways, and two main different mechanisms can be used by these cells, and we can exploit this mechanism in order to develop therapeutic strategies for beta hemoglobinopathies. So in the first mechanism, which is called homologous recombination, basically we can, in theory, we can fix the mutation of a gene. So if this is a gene that in which we have the mutation, such as the beta globin gene, and this is, you can think about that like the sickle cell disease mutation, actually we can provide a donor a piece of DNA containing the wild type beta globin gene, that then will be copied by the cellular machinery into the genome. And so in this way we will have correction of the target gene, in our case the beta globin gene. Then this cut can be also repaired through another mechanism which is called non-homologous enjoining. And actually this is a mechanism that is not very precise. So basically this is a mechanism that we exploit in order to inactivate genes. For example, if we want, in this case, we are targeting this particular gene, for example, a repressor of the fetal gamma globin, and where this gene is cut by our CRISPR-Cas9 or Zinfinger, and then it's repaired. But the repair is not precise, so basically, in this system, the gene is completely inactivated, and this would lead to disruption of this gene. And if you think about this target gene, such as a repressor fetal gamma globin, so we basically, in this way, we inactivate a repressor of fetal gamma globin, and so we reactivate fetal gamma globin. So regarding the first approach, which is based on homologous recombination, when there are several groups who try to correct the beta globin gene mutation, in particular the sickle cell disease mutation, in which basically we deliver in hematopoietic stem cells of sickle cell patient, we deliver our actually genome editing tools, and we deliver the wild type beta globin gene. In this case, this gene is basically copied into the genome, and we achieve targeted correction. But so far, unfortunately, this process of homologous recombination has been shown to be not very efficient in hematopoietic stem cells. So there are several groups that actually reach only up to 3% of corrected beta globin gene, which may not be enough to correct the clinical phenotype. In the second approach, and this is the last approach actually that I'm showing you, is basically aims at increasing the fetal gamma globin gene. So as I told you before, there are several proteins, but even small DNA elements which actually repress the gamma globin gene, the gamma globin fetal gene. And actually, what the idea would be to kill these gamma globin repressors, to inactivate them in order to increase fetal hemoglobin. And this is what we and other groups have done. Basically, we have treated cells coming from sickle cell disease patients with our genome editing tools, and we inactivated the repressor of the gamma globin gene. We were able to achieve increased gamma globin production, and we were also able to show that we and others group were able to show that this increased gamma globin production is able to ameliorate the sickle cell disease phenotype by reducing the percentage of sickling cells. And in conclusion, I just want to say that regarding the efficacy of this approach, actually, as I told you before, correcting the beta globin gene mutation is not still very efficient in hematopoietic stem cells, while increasing fetal gamma globin gene has been shown to be a potential therapeutic approach. Indeed, we show that we can achieve high and curative levels of the endogenous gamma globin gene. Although I have to say that this strategy has not test the active hematopoietic stem cells. So what we have to do now is to ameliorate the efficiency of this genome editing in the hematopoietic stem cells, which are the ones which are long-lasting and will cure the patient. And we will test and improve this genome editing efficiency. And regarding the safety, of course, compared to lentiviral vector, which integrate in several genes in the genome, this is a targeted therapy. So in theory, we are targeting only one specific gene. In our case, for example, in the first approach, the beta globin gene. But actually, this is not completely, may not be completely true. So we have to evaluate the off-target activity of this genome editing tool, because basically this means that some of these genome editing tools can have bind and then cut some unintended targets. And these are studies that have to be done in order to, if this is the case, we have then to improve the specificity of these tools. And finally, just the final remark is that at the end, I think we have to make an effort to compare all these therapeutic strategies which are based on lentiviral vector, genome editing, pharmacological treatment, in order to find the best tool that we can use to ameliorate the phenotype of beta thalassemia and sickle cell disease patient. And finally, this is the last slide. I want to thank you for the attention. And this is the Imaging Institute for Genetic Disease, where I'm working here in Paris. And please feel free to come and visit the institute and the laboratory. Thank you very much.
Organizer to invite me to this workshop on genome editing. I'm going to do an overview of the gene therapy and genome editing technology focus on Duchenne muscular dystrophy. First, some information on Duchenne muscular dystrophy, even if it's unfortunately a famous disease. The Duchenne muscular dystrophy is the most common severe form of childhood muscular dystrophy, affecting 1 to 3,500 male births. DMD is a dystrophinopathy, which is a link inheritance. This disease is due to alteration in the DMD gene coding dystrophy. The life expectancy with ventilatory assistance is superior today to 36 years, and there is no actual cure available. So Duchenne muscular dystrophy is a school case mostly because of the genetic feature of this pathology. Indeed, the DMD gene is one of the largest known genes. It's a very, very large gene which is composed of 2.5 megabase. And the transcript of this gene is composed of 79 exons. and more than 14,000 base pairs. So dystrophin is a large protein, and the isoform, skeletal muscle isoform, is the 427 kg isoform. This dystrophin is a subsarcolemmal protein, as you can see here, is immunofluorescence on muscle section. And you can here see the fibers and the dystrophin labeling. And here it's a reconstitution of 3D fibers, when you can see here the localization of the dystrophin. So the structure of the protein is quite well known, with an N-terminal part, which is an actin binding site. Here a large middle part, which is composed of 24 spectrin repeats, which are called Rho domain, and a cysteine-rich domain here, and a C-terminal domain. And this dystrophin here belongs to a large complex of protein, which allows the connection between the extracellular matrix and the actin binding here, and the cytoskeleton, and all this complex is needed to keep the integrity of the fibers during the contraction process. So alteration in the DMD gene coding dystrophin leads to dystrophinopathies, the DMD and the Becker muscular dystrophy, BMD. It's a disease affecting skeletal muscles, and it's for the two dystrophinopathy, it's mutation in DMD gene, but for the DMD, mutation induce outframe mRNA, meaning that there is no protein expressed. And while the mutation for the BMD are allowed in frame mRNA, and you have a truncated dystrophin expressed. And the phenotype are not the same. The DMD is severe phenotype, lethal, and the BMD is moderate to severe phenotypes. This observation was the rationale for the development of therapeutic strategy of exon skipping, because we can have a truncated dystrophin express, and you can have moderate to severe phenotypes. Exon skipping strategy has been developed on this rationale. And I'm just going to do an overview of therapeutic approach today that are in clinical trial. So the first one is RNA repair exon skipping. And just I want to do a little explanation with for the principle of exon skipping, giving you an example. If you have a deletion of exons 45 to 50 in DMD gene, you have here the outframe mRNA expression, meaning here that you cannot have dystrophin, so you have a DMD phenotype. The principle of exon skipping is to remove one exon, to restore the open reading frame in the mRNA, allowing expression of truncated dystrophin. And this is the development of the exon 51 by Sarepta. This treatment by skipping the exon 51 on the DMD gene with a specific chemistry, PMO, allowed application of this treatment to around 30% of the DMD patient. And for the first time, studies over the 4 last year showed that this treatment prolongs ambulation and change in the rate of declines compared to age-matched control. And this was measured by the 6-minute walk test. And for the first time, the FDA allowed the treatment and approval, the first drug for Duchenne muscular dystrophy. A second treatment has been developed, strategy has been developed, sorry, it's a gene replacement therapy. And as I told you, the gene of the dystrophin and the mRNA of dystrophin is very, very large. So it cannot be packaged in the vector, which is called adeno-associated virus, AAV, because this vector allowed only 4 or 5 kilobases to be packaged. So, as I told you, the full-length cDNA is around 14 kilobases. So, the strategy is to produce mini microdystrophin to be packaged in the vector and to be used in gene therapy emplacement. But the problem is that we have to know which domains are important to the binding of the partners of dystrophin, the function of dystrophin, and it's a long way to establish the best microdystrophin, to be sure that every function and binding site are in this microdystrophin. But there is a clinical trial, that is carried out at the National White Children's Hospital by the Dr. Mendel. Just, it's a phase 1 for the safety. So you have also 2 other approaches, which is drug approach. The first one is the PTC treatment, which is called Translarna. This treatment is in phase 3, and this compound allows to bypass nonsense mutation, point mutation, and allowing to read the mRNA to produce dystrophy. And this drug is in phase 3 today. Another one, it's another approach. It's a drug SMTC, and it's a drug elaborated by Submit. And the goal is to upregulate the utrophin, which is an analog of dystrophin, using drugs. And this approach is being tested in phase 2 clinical trial. So now we are coming to our interest today, novel approach for DMD. So I'm going to show you 3 publications, important, which have been published in Science this year. And these 3 publications showed in vivo use of the CRISPR-Cas9 strategy. So to understand the gene correction by genome editing today in DMD is analogous to exon skipping therapy, which I show you. It's RNA repair. This approach is CRISPR-Cas9 mediated removal of 1 or more exons from the genomic DNA and not the RNA repair, the genomic level. And for that, we have a mouse model, DMD mouse model, and this mouse displayed a premature stop codon in exon 23 of the DMD gene. There is no dystrophin expressed in skeletal muscle of this model. So the goal was to use the CRISPR-Cas9 tool to cut around the exon 23 and to restore to remove it at the genomic level and to allow the reading frame restore here at the mRNA level and to allow the expression of dystrophin. So as I told you, 3 separate laboratories published in Science this year in vivo genome editing with the CRISPR-Cas9 tool. In all these 3 studies, they target the point mutation in exon 23 and with MDX model. I just focus on one of these 3 publications because I can explain after the 2 other ones. So as I told you here, the principle here is that using of CRISPR-Cas9 tool to cut around the exon 23 to remove this exon with the mutation. And the CRISPR-Cas9 with here the guide to target, especially the precise site here, has been delivered with vector AAV, and the three teams choose AAV8 and AAV9 because of the very specificity of this vector to go to skeletal muscle. And they establish two vectors, one with the sequence of the Cas9, like that they can express the Cas9 into the fibers, and another vector with the RNA guide to target specifically around the 23 exon. So two construction, two AAV vector, and they can inject here to the mouse model, and this is the time here, the age of the mouse, and the way of administration. So in this study here, you can see that at the mRNA level, here, the black band is the band corresponding to the removed of the 23 exon. So it's an exon skipping efficiency. You can see here in a section of skeletal muscle, positive dystrophin positive fibers, here and here in red. And they also investigate force in male and female with injected with AAV in intraperitoneal way. And this is MDX force, this is MDX with AAV, and this is a wild type. So you can see here that you have a significant improvement of force. And here you have the summary of the three studies. That this one I showed you, explain you, and the two other one. This is the way of injection of the vector. This is local injection with intramuscular in the three studies. This is systemic injection with retroorbital, or here, or the tail vein in this study. And what we can say that the Cas9 protein used was different for this study and the same for the two other studies. And as I told you, the AAV was 9 for this study and 8 for the two others. And here you can see the result. You have the percentage of modification at the genomic level, which quite low, but when you go to the dystrophin positive fiber investigated by immunofluorescence, you can see here that you have more than 25% of dystrophin positive fiber in skeletal muscle. You have the muscle function here that I show you also, the grip, the strength, the force. You have the cardiac cell editing also studies, which is important, so they can see, they could see cardiac cell editing in the three studies, and you have also the editing in satellite cells, and these cells are very important for the regeneration of muscle. So to summarize these very encouraging studies, each of the three studies demonstrates efficiency using two vector system, AAV CRISPR, and for the guide, they used two different Cas9, and they used also different way of delivery and two different settings. But all showed dystrophin expression at skeletal muscle level, vascular smooth muscle, and cardiomyocyte, and also functional recovery with increased in grid test, with improvement in force generation, in resistance against eccentric constriction, a reduced of serum creatine kinase, and improvement of cardiac function. but there is also the dark side. The dark side for the genome editing is that the concern of the off-target effect. So in these 3 studies, they explore the off-target effect by deep sequencing of the guide target site, and they specifically give attention to the top 10 predict possibilities. And in these 3 studies, none showed evidence of off-target gene editing. Of course, it depends of the guide that you give and that you give to the cell to rescue the dystrophy. And here in the 3 study, it was the same guide. So, okay, there is no off-target, but you have to be very concerned about that. And very recent publication point the fact that you are coming with a Cas9 protein expression, and it's a bacterial protein, and also that you are going with AAV vector. So in this publication, they focus on the host response of these 2 parameters, and they can show that the Cas9 evokes cellular immune response, while the AAV9 does not. That the Cas9 evokes humoral immune response, and that the AAV CRISPR-Cas9 does not evoke extensive cellular damage. So it's also an important point that we have to be concerned about. The CRISPR-Cas9 system is also a very, very good tool to develop animal models to test the therapeutic strategy. And for Duchenne muscular dystrophy, we need models. We have the LDX model, which is not really very affected in terms of skeletal muscle pathophysiology. And we have the dog model, the GMG model, which is very expensive and very heavy to have in the laboratory. So we need GMG model, but also we need Becker model. Because, as I told you, way of investigation on the microdystrophin gene transfer is very important, because you can treat all the Duchenne patients if you can restore a microdystrophin. It's very important to establish which domains is important and is needed to keep the function of the dystrophy. And we have natural microdystrophin or mini-dystrophin in the Becker model. So we can learn a lot of Becker models, and I think that the CRISPR-Cas9 tool is good tool to develop the animal model. And here, very recent paper showed porcine zygote injection of CRISPR-Cas9 resulting in a DMD-modified pig with muscular dystrophy. And this model showed disorder like the DMD human phenotype. So this, as I told you, is important to mimic also the rare DMD cases. For example, duplication of exons or point mutation in 5' at the beginning of gene or at the end of the gene, or in intronic also mutations that impact on the splicing, for example, of the mRNA. But also to mimic the BMD cases, which is very important to study the function and the important domain of the dystrophy, to be sure that when we are going to construct a microdystrophy, it's going to be a good one.
So thank you very much again for the organizers to invite me to this presentation and to this workshop. Is that better? Okay. Please do remind me if the microphone travels away from my face. So I'm going to talk about gene therapy and gene editing for retinitis pigmentosa. But to give you a bit of a background first on what retinitis pigmentosa is for those who are less familiar, I'm going to present just a brief background on this disease, which is a genetically caused disorder leading to blindness. Then I'm going to briefly go on to explain gene therapy for these eye disorders, move over to gene editing for these eye disorders, and then give you an outlook and a bit of science fiction on these technologies. So obviously most of us know that we have two eyes, and the eyes have an inner lining of light sensing tissue. Those are so-called photoreceptors. They are responsible for receiving the image through the lens and so forth, and then conveying that information to the brain. Okay, so you can see the inner lining, that's this orange bit, and then you have this yellow bit over here, that's the optic nerve, where all the information is collected, and then there's a bundle of neurofibers going to the brain, transporting that information to the visual cortex. Now, this light sensing tissue is called retina, and the retina has different things, different layers of cells, and the most important bit, or the other you can forget about at the moment, are these cells. Those are the photoreceptors. Okay, so those are specialized neurons, rods and cones, that detect the photons coming from the environment and transfer that into electric potential to move into the brain so you can perceive an image. Retinitis pigmentosa, and indeed other forms of hereditary retinal disorders, are basically a progressive loss of these light sensing cells. So they continue to die. And not surprisingly, if they're gone, you're blind. Clinically, what we see is this is a normal fundus. So if you look through the pupil into the back of the eye, this is what you see in a healthy individual. And then you have different forms of hereditary retinal disorders. And retinitis pigmentosa is this example, where you have these black pigment clumps that look like the lining of the bone, and that's basically where the name comes from, pigmentosa, and bone spicule pigmentations. That's a classic sign of retinitis pigmentosa. Okay, so what happens in progressive loss of disease? Your peripheral visual field shrinks down, and then you continuously become blind, but that really depends on what kind of retinitis pigmentosa you have. This is an overview indicating that there are many more forms of hereditary retinal disorders besides retinitis pigmentosa. Retinitis pigmentosa is about 50% of all the hereditary forms leading to blindness of some sort. And there are different ways to classify those. I don't want to bore you with that, but just show you that there are different forms. So this shows you the progression with age, right? So some forms of retinitis pigmentosa are very progressive. Some are a bit more stationary. There are forms of retinitis pigmentosa that primarily involve the rod photoreceptors, so a subclass of those light-sensing cells that are responsible to look or to perceive very dim light stimuli. So if they are gone, you can't really see anything in this corner of the room. Maybe more in the back where there is still some bright light. And then there are other forms, but I won't go into detail here. So 50% of all those genetically determined blinding disorders are called retinitis pigmentosa. Now, retinitis pigmentosa in itself is fairly rare. There's still about 200,000 people in Europe, so a considerable number of people. There is a bit of a difficulty with this term again, because retinitis pigmentosa is really a multitude of different disorders that share, well, an historic similarity. Historic with meaning that 100 years ago people said, oh, this is retinitis pigmentosa, without having the means to identify the genetic cause, the pathomechanism of the disease, and so forth. So it's a bit of an umbrella term lumping a lot of things together. Accordingly, the symptoms vary quite widely, and as I pointed out, some are more progressive than others. Some focus on the rod photoreceptors, which are there to detect dim light stimuli. Some affect more early on or more at all cone photoreceptors, which are responsible for daylight vision, color perception, and high visual acuity. And then one can say that the absolute vast majority of patients need visual aids during their life, which often concerns very poor visual acuity and so forth, leading to blindness. Now, from a genetic level, there is a multitude of causes for retinitis pigmentosa, and these are acronyms indicating autosomal recessive inheritance patterns, right? So both alleles need to be affected in order for the phenotype to develop. 20% are autosomal dominant, which means that one allele has to be affected and bang, you have the phenotype. And that's going to be very important later on for gene therapy and gene editing. And then 10% are X-linked, right? So I am, for example, one of the species that has only one X, and then the ladies in the room have two X. So ladies are typically not affected in an X-linked disorder, but there are, of course, exceptions to the rule, as always in biology. And then 40% are simplex cases. What does that mean? Well, it means that a patient comes to us in the clinic, says, oh, nobody in my family ever had such a thing, and we discover a gene defect. So we're not entirely sure whether that gene defect, especially if it hasn't been described before, is actually recessive or dominant. Well, we can kind of understand this from genetic analysis sometimes, but we can't be absolutely sure at times. Okay, so there's a multitude of genes that can lead to retinitis pigmentosa, and each gene by itself has the option of being mutated at various places. So that adds up to a good number of mutations that we can deal with and work with. That's a positive way of thinking about that. Now, this is a picture I've shown you previously, so the eye and how it looks like if you have this disease. This is from Anneke de Hollander from Nijmegen, one of the centers in Europe doing genetics. And she has this nice graph showing you the heterogeneity of the genetic background of retinitis pigmentosa. So this is just picking up the recessive forms, right? We're not talking about the dominant forms or X-linked. And here you can all see that in 2010, 50% were still unknown. Now with next generation sequencing and so forth, this has gone down, but it's clearly not zero, right? So, and we already have 200 genes available. And you can also see that the pieces of the cake are fairly small, right? So even if you went ahead and treated all patients with USHA2A, which is a good chunk of the cake, you still only figured out a therapy for, let's say, 1, 2, maximum 5% of the population that actually has this kind of disorder. So that's a challenge. Right, so the take-home message from this first part really is that we have a considerable unmet medical need. We have people going blind, and 200,000 people in Europe is not, it's quite a bit. We have a large number of different genes and disease types, so keep that in mind. We have a progressive degeneration. It's tissue that is post-mitotic, meaning that it doesn't regenerate. We don't have what the muscle people have, the satellite cells, and we don't have the bone marrow, right? So whatever neurons we lose in the eye is lost. The only thing we can do then is stem cell transplantation or electronic implants or that kind of thing. But gene therapy only works as long as the cells are still there and viable, meaning they could potentially work as they're supposed to do. So, positively speaking, we have an extremely attractive organ for gene therapy and gene editing. It's small. We have 2 of them. Fairly easy access. So 2 of them, it's a laughing matter to some, but actually it's quite important. We have internal control. A lot of the genetic diseases are quite variable because we don't understand all the epigenetics and whatnot. So having an internal control is a huge benefit for a clinical trial. Easy access, it's out in the open. We don't have to cut, the belly open or something like that. We have excellent monitoring ability, so cellular resolution, structural imaging, electrophysiology, non-invasive electrophysiology, and so forth. We have a so-called immune privilege. That's also important. For example, sticking in a bacterial enzyme such as Cas9. You don't necessarily want this out in the open. Now, immune privilege is also a bit of an iffy term. After all, there are diseases with autoimmunity in the eye, uveitis, you might have heard of that. So immune privilege is a concept that clearly is there, but it's not an absolute hermetically sealed organ that you can do whatever and nothing happens. Especially if you start with sticking a needle inside, the barrier is actually broken by definition. Internal control, I went ahead on that, and there's a wealth of scientific literature and knowledge about the eye and the pathomechanisms and so forth. We've heard from previous speakers already that gene therapy is basically a means to bring a therapeutic gene into the affected cells, right? So you have a therapeutic nucleic acid, like the RNA or DNA, that holds the correct information of the gene that is affected, carries a mutation in the cells, and because of the mutation in the cells die, and you basically bring in the correct version and prevent the cells from dying, but rather want the cells to function properly. Okay? Very simple. Now what you use is, for example, some kind of vector, typically a viral vector, such as adeno-associated virus. You could, for example, also use lentivirus or adenovirus, but in the eye, AAV has shown tremendous benefits. Okay, so AAV is a single-stranded DNA carrying molecule. It's really teeny tiny. It's basically a nanoparticle, you could say. The outer dimensions are 20 nanometers. Really, really teeny tiny. And then that also makes understandable why there's only so much space inside. So the packaging capacity is quite limited. But fortunately, a lot of genes that affect the eye, in terms of retinitis pigmentosa, actually do fit in there. So what you do is you stick the therapeutic gene in there. You have front bits and back bits of viral DNA that help the process. And then this actually acts as a taxi to bring the therapeutic gene into the target tissue. And then the DNA is in there and can basically substitute the disease-causing gene and act like a healthy gene. So it stays in the cell, but it's not integrating into the DNA, and that's important for safety issues. Now we've discussed this just previously in the previous speaker, right? So diluting out the therapeutic gene. Now remind yourself again that the cells in the eye are post-mitotic. There is no mitosis, there is no dilution. So in effect, a non-integrating vector is fantastic for the eye. We have no issues with insertional mutagenesis. There's no disruption of any viable genes. The genes are there, but are not diluted, so bingo. So this is an example. You have a single nucleotide that is orange instead of blue, meaning that there is something wrong. And this is a patient with choroidynia. Oh, sorry, even I have probably pronouncing it choroidynia. So choroidynia is one of the genetically determined eye disorders. People get blind. And for those of you who don't understand the genomic gibberish here, this is the coding sequence, and then you have a change from a C to a T at this position, and then that ends up being a stop mutation where the protein isn't produced properly, and the protein isn't there essentially for functioning, and the cells suffer and die. So this is an example of a retina that looks nice and orderly, and if you have the mutation, then it all goes haywire, right? You don't want this. So what you do with gene therapy is basically you use these nanoparticles, you stick in the, sorry, this is in German, embarrassing. You stick in the gene that is non-mutated, and then you bring the retina back in order. Ooh, 5 minutes. That's going to be a challenge. So I'll skip ahead. So this is obviously this is an example of a patient. Everything is degenerated. This is the only bit left for him. That was 2010. It melts away 4 years down the line. 2 years down the line, even less available. We did gene therapy, and this is how we do it. This short video, if it works, where you see the surgery happening. So close your eyes for 10 seconds if you feel queasy. So you enter the eye through 3 ports. Now you can look again. You put away with the jelly inside the eye. You don't really need that. It's vitreous body. And then what happens is you inject the viral solution right at the site where you want to transduce the cells, namely the subretinal space. And I'll try to skip forward for time reasons. So this is a blip, and then you increase the injection of the viral vector. So these are single cells here almost with photoreceptors. So this is live imaging or cellular imaging while we inject it at the right place. And then the results, this is unpublished data from this trial, shows that 3 months down the line, patients can see this stimulus, blue, that they haven't seen before, and this is color vision. So this is an example of achromatopsia gene therapy, where people don't have any function in their cone photoreceptors, they don't see color, they don't see, they can't deal with bright light and so forth, and we've been able to change that in these patients. So what we do in Tübingen at the moment is gene therapy for choroidemia, gene therapy for achromatopsia based on CNG-A3 mutations, and we aim to give them back the color vision and high visual acuity and reduce their glare problems. And we're aiming to develop a gene therapy for PD6A mutations that should start next year. RPGR-linked retinitis pigmentosa is a clear target, as well as BEST1 and retinoschisis, juvenile retinoschisis. Now, this is also already quite a commitment. It's several $1 million and a large team behind that objective. But we're not alone. There are plenty of centers targeting hereditary retinal disorders, and you can see a map here. Now for gene editing, I'm a bit pressed for time, so I'm wondering whether I should show this movie. Has anybody still quite... Yes? Okay.
Like movies.
Right, so you can't hear the audio, but what you see, this is a Cas molecule, right? So this is why Cas was developed by evolution. You have this phage infecting a bacteria. The phage then brings in his own DNA, and the bacteria obviously doesn't like that new information. So what the bacteria does is has already developed, so that's skipping quite a bit of the system, already has developed an RNA that fits exactly the RNA, the DNA of the phage. It basically prepares the Cas system to take the phage DNA, and then it binds it, cuts it, and basically destroys it, right? So the phage DNA is gone. So it's a defense mechanism. So now people realize that, hey, wait a minute, we can use this. We can use this for our purposes. So all we need to do is we have to figure out how to, bring this guide molecule in the cell. And then if we find the right guide molecule, we can tamper with the DNA in the cell, right? So we can tell the system, okay, please cut exactly there, where the DNA is homologous to the sequence that we put into the cell. And then the cell has a repair mechanism that was indicated by non-homologous recombination earlier in the talks. So it finds ways to bring them back together, but it's error prone, and the red indicated that it's mutated in a lot of times. So you can basically knock down the gene. So that's fantastic news. at least for autosomal dominant disease. The problem is that these are quite error prone, right? So if you want to properly correct it, it's a bit more difficult. You have to bring in this template DNA, which tells the cell this is how it should be, and then the cell hopefully actually repairs it, right? But this is a bit out of the hand, so that's the difficult bit, right? How do you tell the cell to not make the first, but the second, and to make sure that the cell actually does exactly what you think it hopefully might be doing? And that to 100% specificity. And we've heard already in previous talks that we are successful in that. Principally, yes, but to what extent? 1, 2, 3, 4, 5%? And what happens to the rest? And what happens to off-target effects and if things go wrong? So the key differences, I guess, between all those really nice shiny papers in Science, Cell, Nature, and so forth, is that typically they use, first of all, cell culture. Cell culture are very clean systems of dividing cells. The retina is neither of those. The retina is neural tissue that doesn't divide, that doesn't undergo mitosis. First of all, it's challenging to deliver it, right? So you have 3 compounds that you deliver at the same time into the same cell. So in cell culture, you just throw some chemicals on, and the cells will take it up. It's called transfection. Very easy compared to doing surgery, bringing in exactly the right amount of the information, the bacterial enzyme, the guide RNA, and the template DNA. Not easy. No active cell cycle. So the repair mechanisms are actually happening in the SMG2 phase. You'd actually have these in the retina because they're not dividing. So there is very limited repair mechanisms, and there is no way so far to really say, please do HDA rather than the error-prone mechanism. And the DNA is less accessible. Usually the photoreceptors are busy catching photons and creating action potentials. They have limited the availability of the DNA. Now, what I think CRISPR-Cas editing would be great is for autosomal dominant mutations, where you can knock down one gene. That's much easier than doing the rest of it in the same time. However, you might be limited by the repair mechanisms that I pointed out. You may benefit from addition of a healthy copy of the gene, and the off-target effects are problematic. In previous papers, they showed the risk is about 1 in 1,000. So I'm very surprised that the previous speaker mentioned that there is no off-target effects being found in that case. Now, with autosomal recessive mutations, it's much more tricky, right? So, first of all, you have different mutations for each gene. So you have something in the range of several thousands of guidance RNA molecules and template DNA molecules that you have to develop, right? Each individual sequence would be looked at by regulators as a new agent, a new new drug, investigation of a new drug. So this is a very conservative figure of 5 million in 5 years you need to develop every single one of them. So multiply that by several thousands and you know the challenge we're looking at. And then the remaining risk of mutagenesis. And one paper indicated 50% rate of that. Now, I'm not quite sure whether I would like my patients to have a 50% of mutagenesis in the eye. Now, if I may, just 2 examples. This is, to my understanding, the first proper test run for gene editing actually in the eye. So what they've done is they used mice. They used a special mouse that is green. So if you take the mouse and put blue light on it, it actually glows in the dark. Pretty cool. Now what they try to do is basically knock out that green glow, right? And what they used is just CRISPR-Cas. They targeted that glow gene. And the result, as you can see, this is fairly green, and this is not so green. So they were fairly effective, right? So this bar graph shows that if they use the right, thing, the right combination of tools, they cut down the green to only 15%. So that would mean that they were actually hugely effective, 80 to 90% effective of actually doing what they intended to do. Fantastic. what they did is they used the, what you know from gene therapy, the AAV2. They had one that coded for the bacterial enzyme Cas9 and one that encoded for the single guide RNA, but they didn't have the, they didn't correct for anything. They just knocked down stuff, right? But that's pretty impressive. When I showed that to a colleague, he said that he would only believe the data when he falsified it himself. But I mean, these are fantastic scientists, and I really hope that we're right. So this is another study. This is principle of gene editing for a model of RP, a model for retinitis pigmentosa. So here they knocked down a dominant gene. So what they did was, they didn't go into the eye, unfortunately, but what they did, they took zygotes, right, mouse zygotes, and then they put in the protein, the Cas9, they actually injected it into the cell with like superfine needles. And then they injected also the guide RNA and the template. Sorry, this is actually not knocked down, so this is actually, editing them. So they actually modified the sequence to be the correct one, right? So this is the next step. But they did it in zygotes, not in the eyes. This is a bit complicated, but for all non-molecular biologists, the most important bit is down here. So this is looking at the efficiency, right? So they did this to the zygotes, then used the ones that survived the procedure, find the words, that survived the procedure and implanted those. So they had already a selection. They don't say in the paper how many survived. They used those, and then from those they created offspring. Now 14% were still where they were, so nothing had happened. They were still basically having RP. 35% actually had the correction. That's pretty good. Not 85, but 35. But 21 plus 28, so altogether 50%, actually showed deletions of one or multiple base pairs. Now, that's not ideal, right? Not for going into a clinical trial. Now, I'm not saying that this is the end of CRISPR-Cas, clearly not. But I'm saying that we are still a bit away from developing something that will help a majority of our patients. So, outlook in science fiction, I think dominant disease will potentially be treated by CRISPR-Cas, because knocking down is much easier than actually correcting for it in a very precise way. I think autosomal recessive and X-linked, which is also mostly recessive. I think gene therapy, by bringing in the correct gene sequence, is still the way to go, because you can bring in one gene and correct for all the mutations, treat all the patients with mutations at different places of that gene with one drug. That's a huge benefit. I think there's quite a bit of hype around gene editing, and I think that will die down after a couple of years of disappointments and success and disappointments and success. And there will be gene editing tomorrow, and I think then we will probably be more ready to go into patients and help them. And I think in the next years there's going to be a huge change that affects not only medicine but agriculture and the whole ecosystem. And one example is obviously livestock. They're already editing cows not to have horns. They're editing pigs not to have certain diseases and so forth. So this is going to be the biggest impact on our life rather than medicine for now. But it's very good to see that people from the National Council of Bioethics, European Union, this group, and others really take their time to discuss what is happening now, how do we deal with that, and that's going to be really important. And with that, I would like to acknowledge all the colleagues that fed into this work in Tübingen and other places, and I would like to answer questions.
Thanking the organizers of the meeting for inviting me to be here today. As it has been said, I've been working in assisted reproduction and I've been working in stem cells. And I have to say that even though I'm very interested in genome editing in gametes and in embryos, I'm not an expert, technically speaking, in the techniques of gene editing. So if there are any precise technical questions, I might not be able to reply to them. So let's see what we can take out of this presentation. My presentation is about genome editing in the germline. I will cover both the male and the female germ cells to be edited. I will talk about experimental genome editing in embryos, also what has been done so far in human embryo genome editing. There are 2 papers already dealing with this. I will cover what are the indications for genome editing in human reproduction, and I will finish by mentioning certain issues about genome editing as a tool for fundamental research, and I will finish by giving you my own conclusions. The first thing I want to mention is that most of what I will cover in my presentation is about CRISPR-Cas system, but maybe the same general things could be related to other techniques of genome editing, such as the one that have already been mentioned, such as zinc finger nucleases or talents. But in fact, it has been the incorporation of this fantastic technique, which is a CRISPR-Cas system, that has made the field evolve so rapidly, and certain questions have arisen due to the fact that this technique has come to the possibility to be incorporated in many labs. It's a much, I would say, easier technique to be applied. It's less expensive, and in a way it has facilitated the incorporation into many different possibilities and many different areas of knowledge. Let me briefly mention that, in fact, this technique, and this has already been said by the previous speakers, this technique was put in place in the early 2000s. In fact, it was a Spanish scientist who described that for the first time as a mechanism that bacteria had to try to protect themselves from viral infections. Later on, it was different groups, like the group of Moineau and Charpentier, described and defined the critical components of the system. And it was later on that Charpentier, Doudna, and Signis that confirmed the results that were made by these previous researchers. And it was in 2013 then Zhang and Church described how this system could be used in mammalian cells, and this is one of the first examples on how this technique has been incorporated in many mammalian cells that we will see and we have seen during this morning presentations. The mechanism, again, not trying to overlap with other presentations, but the main idea is to create a double-strand break. This can be done, as I said before, by different means, using different systems, meganucleases, zinc finger nucleases, talents, and now CRISPR-Cas system, which, as I said, is a much simpler, less expensive, and a more usable system in many labs. And the second thing we need to do is to fixate the cell repair by cell repair mechanisms, and this has already been mentioned before. These mechanisms correspond to non-homologous enjoining and to homology-directed repair. I'm not going into much detail into that, but this is the way genome editing works and CRISPR-Cas works. Again, some schematic representation, and the main thing we need to consider is that we can inhibit certain genes and we can activate certain genes by the use of this nice system. And if you want to read more about this, there have been tons of bibliography that has been produced, and this is a review that was made last March in Nature, called Riding the CRISPR Wave, in which you can have a lot of information about this system. Going now straight to germline genome editing. DNA editing of the germline can be performed in any cell or tissue corresponding to the germline, as the name says. It can be done in gametes, such as oocytes or sperm. we will see how this can be done. It can be done in zygotes or embryos, and it can be done in stem cells if these stem cells are going to be used to produce gametes to further give rise to an embryo. So in all these cases, if we edit these structures, these cells or these tissues, we are going to edit the germline, and this will have consequences for the offspring that will be produced through these different cells or these gametes. Let's go into more detail onto this. With respect to oocytes and sperm, the problem is that we need to have the CRISPR-Cas system work. We need that the cell divides, cleaves, and if we have an oocyte, this oocyte by definition is already a mature oocyte, so it's not going to react to what we are going to do unless it's going to have a further division. What we can do is to try to work on stem cells, and this is the approach that has been done in most of the cases for spermatogonial stem cells. So for sperm, what we will do, we will be able to use spermatogonial stem cells that we will be able to edit, and as a consequence, the sperm that will be derived from these cells will be edited and will have the genome changed. The problem is with the oocyte. The oocyte is more easily accessible for genetic manipulations, but the limitations are due to the low numbers and the low efficiency of the technique. And also we can consider that IVM needs to be considered in that case, and trying to get the changes done in the oocytes is something that is still a big challenge for this technique. Going to some examples, let's go first for stem cell genome editing. And what I am bringing is some of the publications. This is a publication of 2014, and in that case, these authors published genome editing in the mouse, in mouse spermatogonial stem cells, and in that case they used zinc finger nucleases. The same thing could have been done with CRISPR-Cas, but in that case they used to simulate homologous recombination in spermatogonial stem cells. What they got is the correction of the cells, and these cells maintain several properties of spermatogonial stem cells, and in fact this was one of the first papers that showed the proof of concept for genome editing in germline cells, in that case in the mouse model. Another model that has been used for spermatogonial stem cells is the rat model. In that case, they used CRISPR-Cas to target heritable mutations within rat spermatogonia. They use this system, and they showed that by using this system in spermatogonial stem cells, they prevented animal mosaicism, because if you do this manipulation in zygote or in blastocyst, you can have, as you can see in the slide, some heritable and non-heritable mutations, and at the end, the animal that is being produced is a mosaic, and you have to purify this line to try to get rid of the non-heritable mutations and the one that are constituted by off-target mutations. Moving towards embryo genome editing. Now we move to the embryo itself, to the zygote, to the blastocyst. And it has to be said that the highest efficiency is expected in one cell stage, zygotes. If you modify only one cell, the progeny of that cell would supposedly have all the genome edited in all the cells that come from this single cell. The high efficiency has been observed to produce both single mutants and double mutants using direct injection of Cas9 mRNA and sgRNA into zygotes. And let's see what has been done so far. First, in animal models. Here we have a paper that has been published this year by a Japanese group that shows the generation of a non-human primate model of severe combined immunodeficiency using highly efficient genome editing. It has already been said by the previous speakers that this CRISPR-Cas system is a very good model to try to model certain diseases in animals. human diseases in animals by using this system, trying to incorporate these mutations into the animal models. And what these authors have proved is that they can generate immunodeficient non-human primate model by editing fibroblasts and embryos. They have obtained good results with reduced mosaicism, and they show that the marmoset phenotypes were similar to that of human ex-SCID patients. So, in fact, they have proved that by using this system, they have a good model for this severe combined immunodeficiency disease. Another thing that has already been done in the monkey, in the cynomolgus monkey, via Cas9 RNA-mediated gene targeting. In this case, what they have used is one cell stage cynomolgus monkey embryos, and they have disrupted simultaneously two target genes, the ones that you see on the slide, in one step, and they have not observed any off-target mutagenesis by comprehensive analysis. One of the main problems that you can observe by using this system in single cells are off-target mutations. Apart from having the mutation that you want to address in the specific cell, you have other mutations that arise at the same time and that are, I would say, collateral damage caused by this technique. By moving on, other models that have been used, and this has been done in the mouse. These authors, and this is a paper of 2014, they have shown that they can prevent muscular dystrophy in mice by using this system, CRISPR-Cas system of the germline DNA. They have corrected the dystrophin gene. This has been addressed in previous presentation. They have produced genetically mosaic animals containing a wide range of correction of the muscular dystrophy gene from 2 to 100%. And at the end, they have shown that the degree of muscle phenotypic rescue in mosaic mice exceeded the efficiency of gene correction, probably reflecting an advantage of the corrected cells and their contribution to regenerating muscle. Let's move to what has been done to human embryos. As I mentioned before, there are two papers published this year that have shown that it is possible to modify the human genome in human embryos. In the two cases, the authors have used tripronucleate eggs, so zygotes, in fact, that have been abnormally fertilized and that are of no use for the clinic. They cannot be used to be transferred to the patients. And the first paper that was published, in that case, they used the CRISPR-Cas system to cleave the endogenous beta-globin gene. They showed that the efficiency was low and the edited embryos were mosaic, as it could be expected. Off-target cleavage was also apparent in these tripronuclear zygotes, and the repair of the beta-globin gene locus occurs preferentially through the non-crossover pathway, the two systems. This is the one that is being used in that model, and that if we need to really apply this methodology, we need to improve the fidelity and specificity of this platform, which is a prerequisite for any clinical application of this system in the human. This will be covered in the next presentation, but as you can be all aware, there has been a big discussion and a big debate around the possibility to modify or to edit the genome of human embryos. There have been extensive discussion all over. There has been the publication of many statements by different scientific societies, by different bioethics committees. This will be covered by Anneli later on. I think that these two papers were the ones that at the end represented the starting point of all this discussion, when we all realized that this could be done efficiently or not, but in the human, to try to modify the genome efficiently in human embryos. The second paper, same situation, the use of 3PN embryos. In that case, they successfully introduced one allele in these 3PN embryos, and they showed that even though some improvements were needed to try to increase the efficiency and the safety of the system, they could really manipulate these human embryos, and they could really edit the human genome of these embryos that, again, were embryos that were of no use for human reproduction or for the patients, in any case. The limitations of human embryo genome editing. At the end, the use of genome editing in human embryos will require the use of pre-implantation genetic diagnosis in order to identify the modified embryos from the non-modified. Once we perform the technique in these embryos, we need to really be sure that this has been done appropriately before we transfer these embryos to the patients. Another important thing that we need to mention is that we need to sequence the full genome of the embryo to know exactly what has been done. And the third thing to be highlighted is that the generation of chimera or mosaic embryos as a result of inefficient nuclease cutting or inaccurate DNA repair is something that we need to address, and in many circumstances, this generation of chimera and mosaic embryos will remain undetected. This paper was produced by a group of people in the European Society of Human Reproduction and Embryology, a society in which I belong to and I have been involved for many years. And we produced that paper referring to CRISPR-Cas to address the issues that this system implies for human reproduction. So this is a review of critical, technical and ethical issues that should deter from employing this system in human reproduction. What are the indications? In these cases that I will not go through due to the lack of time, in autosomal recessive disease and autosomal dominant disease, PGD, so pre-implantation genetic diagnosis, is the technique of choice if we want to avoid the birth of children affected for a certain disease. There's no point in trying to use such a complex technique such as gene editing for these cases. We have a very good technique, which is PGD, that gives excellent results and excellent I would say, results in terms of pregnancies, so there's no point in trying to use genome editing for these cases. But in other cases, such as the one that are listed here, the patients, the parents, both affected by the same monogenic disease, example cystic fibrosis, one member of the couple being homozygous for an autosomal dominant disease, another example, or one member of the couple affected by a chromosomal structural aberration. In those cases, genome editing could be an indication. And you see what needs to be done in the three cases to try to correct the affected gene in the germline of one of the parents or in the embryos that the two parents produce. Other applications can be constituted by common diseases, like, for example, heart disease, or cancer, or diabetes, or multiple sclerosis, but the problem is that in those cases, the heritable influence is polygenic, so we will have to modify more than one gene, and I would say that at this time, this is not even to be considered. A more distant frontier would be to reshape non-medical traits. There is a lot of discussion to be done for this. We can also envisage to correct genes related to infertility and to try to avoid the continuity of this fertility. And we can also eliminate mitochondrial DNA mutations, which in fact has already been done by genome editing, as this has been published last year by the group of Belmonte. In fundamental research, there needs to be an improvement of genome editing technique itself on the different types of target genes. We need to really improve and optimize which are the best cells to use to perform this technique with good results. This technique is a tool to study fundamental questions of human and animal developmental biology, and you can see here as an example this system as a tool for the functional study of genes involved in pre-implantation embryo development. And this is one of the areas that is being you try to switch off a certain gene that you think could be involved in implantation or in embryo development by using CRISPR-Cas system, and if it's the case, then the embryo will not develop or will not implant, and you will ascertain what is the function, the real function of this specific gene. As a conclusion, I would say that genome editing is an excellent tool for the generation of disease models as well as for the study of genes involved in pre-implantation and post-implantation development. I would say that genome editing cannot be actually performed with sufficient precision to allow scientists to responsibly contemplate creating genetically modified babies. The problems are inaccurate editing and off-target mutations. And research in the human germline, including gametes and embryos and stem cells, should be promoted to determine the usefulness and the safety of the technique before we consider the possibility to apply it clinically. Thank you for your attention.